Depleted uranium effects on plants and soil microbiome. Depleted uranium poses significant risks to plant health, influencing both morphology and metabolic processes. Research indicates that exposure to depleted uranium can lead to various detrimental effects on plants, including reduced growth, changes in physiological functions, and alterations in nutrient uptake. Morphological effects can be directly observed via growth inhibition. Depleted uranium exposure has been associated with significant reductions in plant yield, shoot growth, and root development. Studies have shown that plants exposed to uranium exhibit stunted growth and reduced biomass accumulation compared to the control groups. Morphological anomalies have also been documented in plants subjected to depleted uranium. These can include abnormal leaf shapes, discoloration, and overall deformities in plant structure. Such changes can impair the plant's ability to conduct photosynthesis effectively. Continuous contact with depleted uranium can alter the architecture of root systems. Roots become shorter and less branched, limiting their ability to absorb water and nutrients from the soil. The presence of depleted uranium can disrupt photosynthetic activity by affecting chlorophyll content and the overall photosynthesis rate. This impairment is often linked to oxidative stress induced by uranium toxicity. Depleted uranium affects the uptake of essential nutrients by plants. The presence of uranium can interfere with the absorption of macro and micronutrients, leading to deficiencies that further exacerbate growth issues. For instance, the uptake of calcium and magnesium may be particularly affected, which are critical for various physiological functions. Plants exposed to depleted uranium may exhibit increased levels of reactive oxygen species, leading to oxidative stress. This condition triggers various defensive mechanisms within the plant, including the activation of antioxidant enzymes aimed at mitigating damage caused by uranium exposure. Uranium is a well-known toxic metal for plants, and its exposure disrupts various physiological and biochemical processes, including seed germination, root and shoot growth, photosynthesis, and nutrient uptake. Furthermore, uranium toxicity leads to the accumulation of high levels of reactive oxygen species in plants, which causes lipid peroxidation and genotoxic effects. The bioavailability of depleted uranium from the soil is influenced by its chemical form and environmental conditions. Some forms may be readily absorbed by plants, while others remain largely immobile and inert. Some forms, like uranium oxides, are relatively inert and can remain in the soil for extended periods without leaching into groundwater. Other soluble compounds can form complexes with anions and organic matter, thus enhancing their mobility. Chelates in soil enhance uranium uptake by increasing its solubility and bioavailability, facilitating root-to-shoot translocation and interacting with nutrient dynamics such as iron and phosphate homeostasis. These effects are crucial for understanding how to manage uranium contamination in agricultural systems and for developing effective phytoremediation strategies. Factors such as pH, oxidation reduction potential, and the presence of organic matter play crucial roles in determining how uranium interacts with soil. The solubility of uranium in soil is highly dependent on pH levels. At low pH, which indicates acidic conditions, uranium tends to be more soluble and available for plant uptake. Conversely, at higher pH levels, which means alkaline conditions, uranium forms strong insoluble compounds, which significantly reduces its biological mobility and availability. This results in lower uptake by plants as the uranyl ions become less accessible. Soil pH also affects the availability of other essential nutrients like iron and phosphate, which can interact with uranium uptake. At low pH, uranium can displace inactive iron from iron phosphate complexes, potentially increasing the bioavailability of iron while simultaneously enhancing uranium uptake. However, if phosphate is limited, the toxic effects of uranium become more pronounced due to its interference with phosphate homeostasis in plants. While low pH can enhance uranium availability, it can also lead to increased toxicity in plants. High metal bioavailability in acidic soils often results in toxic effects such as chlorosis and stunted growth. Therefore, while depleted uranium may be more readily taken up at lower pH levels, it can simultaneously pose greater risk to plant health. DU penetrator rounds corrode over time, losing about 25% of their mass within 7 years. This corrosion can lead to gradual release of uranium into surrounding soils and water, thus contributing to environmental contamination. It has been proposed that the corrosion of DU materials occur in two stages. First, the oxidation of zero-valent uranium to uranium-4 dioxide, uraninite, and secondly, the oxidation of uraninite to U6 minerals. Estimations based on modeling suggest that it would take 22 to 26 years to fully corrode a DU penetrator in soil, indicating its long-term impact on the environment. Depleted uranium affects the root growth of plants in several significant ways, primarily through its toxicity and interference with essential nutrient uptake. Studies have shown that plants subjected to depleted uranium exhibit stunted root development, which limits their ability to absorb water and nutrients effectively. This inhibition can significantly affect plant health and yield. The presence of depleted uranium in soil can disrupt the uptake of essential nutrients, particularly iron and phosphate. Uranium competes with these nutrients, leading to deficiencies that further impair root growth. For instance, when phosphate is limited, uranium can be absorbed more effectively, exacerbating its toxic effect on root systems. This interference can result in chlorosis, observed as yellowing of leaves, and poor overall plant health. The exposure to depleted uranium can also induce oxidative stress in plants, leading to an increase in reactive oxygen species. Elevated ROS levels can damage root cells and inhibit meristematic cell division, which is crucial for root growth and development. The stress response may also divert energy away from growth processes towards defense mechanisms. DU exposure may also induce morphological changes in plant root systems, such as reduced branching and altered root architecture. These changes can further limit the plant's ability to access nutrients and water from the soil. The chemical form of uranium in the soil affects its bioavailability to plants. In acidic soils, uranium becomes more soluble and available for uptake, potentially increasing its toxic effects on roots. Conversely, in neutral or alkaline conditions, uranium tends to form insoluble compounds, reducing its uptake but still posing risks through long-term soil contamination. Depleted uranium has significant long-term effects on plant metabolism, impacting various physiological and biochemical processes. Nutrient availability and uptake are affected when the plants are exposed to DU over extended periods. This can disrupt the balance of essential nutrients, particularly iron and phosphate. Research indicates that uranium can displace inactive iron from iron phosphate complexes, potentially making active iron available for metabolic processes. However, when phosphate is limited, the toxic effects of depleted uranium become more pronounced, leading to deficiencies in phosphorylated compounds that are crucial for plant metabolism. Long-term exposure to depleted uranium can result in chlorosis due to impaired nutrient uptake. This deficiency affects photosynthesis and overall plant health, as chlorophyll production requires adequate levels of both iron and phosphate. Thus leads to the impairment of photosynthesis. Studies link DU exposure to decreased photosynthetic rates. The presence of uranium can lead to oxidative stress, resulting in increased production of reactive oxygen species. This oxidative stress can damage chloroplasts and reduce the efficiency of photosynthesis, ultimately affecting biomass accumulation and plant growth. Photosynthesis is impaired via DU-induced alteration in chlorophyll content in leaves. Changes in chlorophyll levels can further exacerbate the effects on plant metabolism, leading to reduced energy production. Long-term DU exposure generates oxidative stress due to an imbalance between ROS production and antioxidant defenses. This condition can damage cellular structures, including membranes, proteins, and DNA, leading to impaired metabolic functions. Plants exposed to depleted uranium may activate their antioxidant defense mechanisms to counteract oxidative damage. However, prolonged exposure can overwhelm these mechanisms, leading to cellular damage and compromised metabolic processes. DU presence alters metabolic pathways as well. Prolonged exposure can interfere with mitochondrial function, affecting ATP production and energy metabolism within plant cells. This disruption can lead to reduced growth rates and metabolic efficiency over time. Chronic exposure to depleted uranium has been shown to alter gene expression related to metal metabolism and stress responses. This alteration can affect various metabolic pathways, including those involved in detoxification processes and nutrient assimilation. The final act is the accumulation of toxic compounds. Depleted uranium is absorbed by plants from contaminated soil and can accumulate in various tissues. This bioaccumulation poses risks not only to the plants themselves, but also to herbivores and humans through the food chain. The accumulation of uranium in plant tissues may lead to secondary toxicity when these plants are consumed by animals or humans, raising concerns about food safety and environmental health. Studies have shown that uranium can also have beneficial effects on plant health, particularly under specific conditions related to nutrient availability. Studies on Arabidopsis thaliana revealed that uranium can displace inactive iron from iron phosphate complexes in plant tissues, thereby releasing active iron necessary for various metabolic processes. This mechanism can lead to improved leaf greening and overall plant growth, especially in mutants that struggle with iron absorption. When phosphate is sufficiently available, the addition of uranium can alleviate symptoms of iron deficiency, such as chlorosis. In conditions where phosphate is limited, uranium's bioavailability increases, allowing for more effective absorption by plants. Under these circumstances, uranium may enhance certain physiological processes by facilitating the release of iron, which is critical for chlorophyll synthesis and photosynthesis. This interaction suggests a complex relationship where uranium can act as a facilitator for nutrient dynamics under specific nutrient constraints. The presence of uranium has been shown to improve growth in certain Arabidopsis mutants when phosphate is adequately supplied. This paradoxical effect highlights how uranium, while generally considered toxic, can have beneficial outcomes when nutrient interactions favor its role in enhancing iron availability and supporting metabolic functions. At lower pH levels, for example pH 4.5, uranium uptake is higher, and studies indicate that this can lead to optimized photosynthesis process. Although growth may be inhibited due to energy being diverted toward defense mechanisms against oxidative stress caused by uranium exposure, the efficiency of photosynthesis itself may improve under these conditions. This suggests that plants may adapt their metabolic pathways to utilize absorbed energy more effectively in response to uranium stress. Research indicates that soil contamination with depleted uranium can cause notable shifts in fungal communities. This suggests that DU can selectively favor certain fungal groups over others, potentially disrupting established ecological relationships within the soil microbiome. The presence of depleted uranium influences ectomycorrhizal fungi associated with plants, which play a crucial role in nutrient uptake and plant health. The changes in fungal community composition due to DEU exposure may affect symbiotic relationships between plants and mycorrhizal fungi, potentially impacting plant growth and nutrient acquisition. Soil contamination with depleted uranium can induce stress responses in fungal communities. Fungi are biogeochemically active components of the soil microbiome, and their ability to adapt to heavy metal stress is crucial for maintaining soil health. Presence of depleted uranium may alter metabolic pathways within fungal communities, affecting their function in nutrient cycling and organic matter decomposition. The interaction between DU and soil microorganisms can influence the biocorrosion processes that release bioavailable uranium species into the environment. The presence of certain fungal taxa may enhance or inhibit these processes, thereby affecting the overall mobility of uranium in contaminated soils. Changes in microbial community structure due to DU contamination can have cascading effect on soil health and ecosystem functioning. Fungi are integral to nutrient cycling, organic matter decomposition, and soil structure maintenance. Thus, alterations in their communities may impact overall soil fertility and plant productivity. A study using soil microcosms found that increasing concentrations of DU up to 25,000 ppm resulted in changes to the functional diversity of soil bacteria, as assessed by their ability to utilize different carbon sources. This suggests that certain bacterial taxa may be more resilient or susceptible to uranium contamination, leading to altered community dynamics. Presence of DU has been shown to reduce the functional diversity of soil bacterial communities. This reduction is characterized by a decrease in the number of carbon sources that bacteria can utilize effectively. As a result, the overall metabolic capabilities of the bacterial community may be compromised, affecting essential soil functions such as nutrient cycling and organic matter decomposition. The effects of depleted uranium on the yield and nutrient content of crops can vary significantly depending on several factors, including the concentration of uranium in the soil, the specific crop species, and the availability of other nutrients such as iron and phosphate. Research indicates that high concentrations of depleted uranium, such as 25,000 ppm, can lead to significant reductions in crop yield. For instance, studies have shown that at these levels, plant biomass, fecundity, and long-term survivability are adversely affected. This suggests that excessive uranium can be toxic to plants, leading to stunted growth and lower yields. The accumulation of depleted uranium in plant tissues raises concerns about food safety and human health. As crops absorb uranium from contaminated soils, there is potential for bioaccumulation within the food chain. Understanding how depleted uranium affects not only crop yield but also the nutritional quality of food is essential for assessing its long-term implications for agricultural productivity. Thank you for listening.